Howdy, my name is Adamari Cerda, and my partner Mario Flores Castro and I will be discussing our experimental lab report on the biochemical tests that include oxidase, catalase, nitrate reductase, urea hydrolysis, CAMP, coagulase, and the Kirby Bauer experiments. To begin, the biochemical tests discussed in this report have the general purpose to characterize and identify a given bacterium. The oxidase test identifies bacteria with the cytochrome oxidase enzyme. The catalase test tests for the presence of the catalase enzyme. The nitrate reductase experiment tests an organism's ability to reduce nitrate to nitrite or free nitrogen gas. The urea hydrolysis test looks for an organism's ability to break down urea into ammonia and carbon dioxide. The CAMP test tests for the presence of the CAMP hemolysis factors. The coagulase test tests for the presence of simply coagulase. Last but not least, the Kirby Bauer test tests for an organism's susceptibility to antibiotics. The materials required for the catalase test include the bacterial samples of Staphylococcus aureus, Streptococcus pyogenes, and the positive unknown sample, in this case 201. The procedures call for obtaining a glass slide and placing a bacterial colony from the agar. Then 2 to 3 drops of hydrogen peroxide will be added. Lastly, results and visual changes will be recorded. The oxidase test requires for Escherichia coli, Pseudomonas aeruginosa, and the unknown samples 201 and 101. Performing this test calls for obtaining a sterile cotton swab, swabbing the bacterium, then adding 2 to 3 drops of oxidase reagent onto the swabs. Lastly, results are recorded. The materials required for the Kirby-Bauer test includes the bacterial samples of Staphylococcus aureus, Escherichia coli, and Pseudomonas aeruginosa. The procedures call for creating a bacterial lawn for each bacterium. Then each plate will be divided into 3 sections and labeled with the first letter of each antibiotic. In this case, it is N, O, and B. Lastly, using tweezers, the antibiotic discs will be obtained and dropped onto the corresponding section of the plate. The nitrate reductase test requires Escherichia coli, Pseudomonas aeruginosa, and Erusiopathiae, in addition to the unknown samples, both positive and negative. In this case, it is 101 and 201. Performing this test calls for using a loop to inoculate bacteria into the nitrate broth tube. Then, 1 dropperful of sulfuric acid will be added, in addition to 1 dropperful of a naphthylamine. Lastly, a small amount of zinc will be added into each broth. The catalase test displayed a positive result for the S. aureus sample with the formation of bubbles after the addition of hydrogen peroxide. On the other hand, the catalase test displayed negative results for S. pyogenes and the gram-negative unknown sample 101. These 2 tests ultimately did not display the formation of bubbles after the addition of hydrogen peroxide. The oxidase test displayed a positive result for the gram-positive unknown sample 101, with an evident color change of dark purple. On the other hand, E. coli displayed a negative result with no color change. As for the P. aeruginosa and unknown sample 201, the results should have been negative, and their outcomes may suggest a minor error within the experiment. The Kirby-Bauer test displayed resistant outcomes for P. aeruginosa against the optogene and bezitracin antibiotics, as there was no formation of a ring around the disc. However, P. aeruginosa did display sensitivity against novobiocin with a ring around the disc spanning 2 centimeters. Additionally, P. aureus and E. coli displayed full sensitivity against the optogen, bacitracin, and novobiocin antibiotics. What was also observed are the formation of small rings around certain discs, such as the zero point 0.6 centimeter ring around the bacitracin disc for E. coli. This indicates a variance in susceptibility and effectiveness of the antibiotics. The nitrate reductase test displayed a positive result for E. coli, P. aeruginosa, and the unknown samples 101 and 201. This outcome is accompanied by an intense red color change due to the organism reducing nitrate to nitrite. On the other hand, E. ruseopathia displayed a negative result as it only showed a small pink color change at the bottom of the tube. This indicates that the addition of zinc was the one responsible for reducing nitrate to nitrite.
For the urea test, we streaked a colony of E. coli unknown 101, unknown 201 onto the slant of an urea slanted agar. For the coagulase test, we inoculated each sample of Staphylococcus aureus and Staphylococcus epidermis onto 0.5 milliliters of rabbit plasma. Then we left it to incubate for 48 hours at 37 degrees. For the CAM test, we streaked a line of Staphylococcus aureus at the center of the blood agar plate. Then we streaked three lines of Streptococcus pyogenes above the line. Then another three lines of Streptococcus agalactiae below the S. aureus line. These were the results of our urea test. For E. coli, we saw negative results as the agar remained yellow. This means that there was no hydrolysis of urea. Unknown 101 also showed that there was no hydrolysis, as it also remained yellow. Unknown 201, there was a change of color from the yellow to a pink purple color, and it was present on the top of the pot and on the agar, on the slant agar. This shows a positive for hydrolysis, and since it's from both places, it's 2 plus hydrolysis. For the coagulase test, we saw how there was no coagulation on the sample that contained S. epidermis. That means this is a negative result, and there's no coagulase present. However, with S. aureus, we saw there was coagulation, as it formed a gel-like substance on the plasma. This is a positive result, which means that there is coagulase present. For the CAM test, first we see how there's a line in the middle, that's the S. aureus that caused hemolysis. Then we see other three lines for the S. pyogenes and S. agalactiae. For S. pyogenes, we see a slight arrowhead hemolysis, which means that there is a positive result as there is CAMP factors present and enhance the S. aureus hemolysis. For S. agalactiae, there's no arrow hemolysis, so this is a negative result and there's no CAMP factors present.
Ultimately, the catalase test displayed S. aureus to be positive under the conditions that allowed the organism to degrade and incorporate a carbohydrate to produce acid with bubbles. For the oxidase test, unknown 101 was observed to have a cytochrome oxidase enzyme that allows the transport of electrons to a final electron acceptor, in this case oxygen. The addition of an artificial electron donor allowed enzyme reduction, and therefore the enzyme presence was in the color change observed to be purple and or blue. For the nitrate reductase test, the addition of reagents A and B allowed a reaction that forms nitrite from nitrate, as seen with E. coli, P. aeruginosa, and unknowns. Lastly, the Kirby-Bauer test displayed that P. aeruginosa is resistant against optochin and bacitracin. Ultimately, varying sensitivities were observed and may indicate different levels of effectiveness.
The conclusion for our urea test is that our unknown 201 is able to hydrolyze urea into ammonia and carbon. This means that this bacteria is able to use urea as a carbon and energy source. For our CAMP test, We saw how S. pyogenes is able to produce CAM factors that enhance the hemolysis of S. aureus. This results in hemolysis with an arrow pattern where the two bacteria meet. For our coagulase test, our conclusion was that S. aureus is able to produce a coagulase enzyme, which is why it's able to form clots in blood plasma.